Hi, my name is Al Kim from Washington University School of Medicine in St. Louis, Missouri, and welcome to day 2 of the ACR Convergence 2025 meeting. We've been covering the cellular therapy and T-cell engager abstracts here, which there are over several dozen. I thought I'd be covering a lot more of the clinical trial data, but what's been really striking in my eye has been a lot of the innovation, the preclinical work. And so I wanted to highlight one of these abstracts, abstract 981, called GNTI 350, a CAR-Treg therapy offering durable immune reset with improved safety in the B-cell-driven autoimmune disease. The title is a little bit overstating since all of this work is preclinical, but nevertheless, this is actually a really interesting compound created by a biotech company called Genta Bio in collaboration with the University of Washington. And what this is a CD19 CAR T-cell that has been polarized after activation to also become a Treg. So this combines two really of the newer concepts in CAR T cells. The first is the killing of B cells vis-a-vis interaction with CD19, but also then functionally creating that CAR T cell to become a Treg and provide an immunosuppressive milieu wherever that Treg is. So as I mentioned before, this is all preclinical work. And so they start off with in vitro work demonstrating that, yes, their CAR T cell can kill CD19 positive B cells. That's not a problem. When you co-culture GNTI 350 with B cells, they upregulate their Treg markers, and then they can suppress other T cells that are also within that culture, again, consistent with the product. On top of that, when you combine that with inflammatory conditions that can create a cytokine release syndrome, those conditions are attenuated. And I think this is the really interesting and important point of this compound, why you would want to combine Treg function with a killing function in the CAR T cell. The main direct effect that the company thinks is its advantage is that it will abrogate cytokine release syndrome issues, because a lot of that is going to be coming from the dying B cells within that tissue. So it will be interesting to see whether or not this actually pans out in humans. They do have in vivo data to suggest this is true, so they do see reduction in cytokines such as IL-6 that are responsible for cytokine release syndrome in mice when it depletes B cells. And then they show efficacy data in a mouse model of lupus, the SLE123 mouse model, strongly attenuated disease progression. So, it's interesting. Innovation is everywhere right now at ACR in terms of CAR T cells and T cell engagers. And this is just one example. There are other companies that are trying to alter the signaling of the CAR construct in order to reduce cytokine release syndrome. Here, what they're doing in this particular example is going to give that CAR T cell an additional function. You could argue this may be over-engineering, right? There are other solutions to get around cytokine release syndrome. And there's also additional questions long-term. How long does the Treg actually last for after they differentiate into these Tregs following CD19 stimulation? And the other issue here too is what happens if these CAR T cells that are now Tregs go into irrelevant tissue? Does it immunosuppress? A lot of questions that need to be answered in long-term, either preclinical studies or in first-in-human studies. But again, innovation is everywhere here, and this is just another example.